Now, it's my great pleasure to give the floor to Professor Maritha Kotze.
Thank you very much for the honor to receive this award on behalf of Stellenbosch University and the National Health Laboratory Service in South Africa. The whole purpose of our project is to bridge the gap between research and clinical practice. And we have developed over many years a pathology-supported genetic testing platform where the research and the innovation and the application all comes together. And more recently, a point-of-care testing platform started to use as just an entry point for this whole process to take off. And then eventually, we could then also bring in research with the service delivery in order to learn from the experience. My disclosure is that I'm a co-founder, a director, and a shareholder of a spin-out company of the South African Medical Research Council. The Research Council provides for us a platform where we can do the whole genome sequencing. Our project was part of the first 6 sequences that has been sequenced in South Africa, and the publication has come out on that work. So the whole idea with the MRC bringing this type of infrastructure to us as scientists is to unlock Africa's diverse gene pool, to enable us to perform gene-targeted therapy, timely surgical intervention, and cascade testing in families. The genomic system is really a virtual platform where different points of data collections can come together to provide reports that we could then use in the end also to return results after it has been approved by a group of experts. What really spearheaded this whole process was a grant that we received as part of the South African United Kingdom Newton Collaborative Research Development Program in Precision Medicine. There's a laboratory in the UK, the Laboratory of Government Chemists, that developed this technology, and we were then invited to join in using our pathology-supported genetic testing platform as a clinical interpretation tool and maybe a decision tool to combine it with their expertise. So what then happened is that we developed a BRCA1 and 2 founder mutation test or founder pathogenic variants. That's most common in our population across the different ethnic groups. And then also the second focus was to develop a para-DNA automated pathway panel for detection of cardiovascular disease risk factors. That's actually relevant in most non-communicable diseases. And those were conditions that I've done research with my students and other researchers over many, many years. So basically a whole lifetime of knowledge of different people working together, all towards the same goal. And when this point of care test is not informative, we can then move on, work very closely with the genetic counselors as basically the gateway towards personalized medicine, precision medicine. And what makes these assays so wonderful for me is that we could place them at certain areas within the algorithm for breast cancer where they could really make a difference for patient care. So the focus would be not only on the therapy-related risk, but we need to start to look at the family implications, so familial versus lifestyle-related cancers, and then eventually, if the person already has got cancer, look across the continuum of care. So it's really a multi-assay pathology-supported genetic testing platform that enables us to apply a 3-pronged approach towards the identification of, like I said, pathogenic mutations with familial risk. Then we look at the genetic underpinnings of tumor subtypes, biochemical abnormalities, comorbidities, because all of these are influenced by modifiable environmental factors and they are very important towards risk reduction. Then ultimately to minimize the increased risk of medication side effects or failure due to genetic variation. It all forms part of a learning healthcare system where we then really try to make sure that the patients access the right care pathway according to their clinical problem. So you can see here we also work with medical schemes in pilot programs. This resulted in medical schemes starting to reimburse some of these genetic tests, because otherwise it would not be accessible. And then always the familial lifestyle and therapy-related risk is kept into mind We then develop these non-communicable diseases pathways reports to see if you need to move further. And then on the research level only, we do whole exome and whole genome sequencing at this stage. So it's really about, moving across these different silos. As geneticists, we usually start with familial risk, looking at the family, but we've soon learned that we also need to look at patients that could maybe prevent unnecessary chemotherapy in early stage disease. Combining the prognostic test with a germline test for cancer development is also very helpful. A BRCA2 positive patient, for example, with a hormone receptor positive or luminal type, if we have done molecular subtyping, are increased risk of late recurrence and relapses, so that it is very important for us to identify those subgroups, because in the end, we might be doing a lot of screening, a lot of patients, but there might be one or two that really needs more individualized care. And I think this is important, is that even if we do a research study, we may start with 100 or 200 or thousands of patients. But if we approach this correct, then we could end up with a handful maybe that doesn't fit the usual picture. They do not respond to the treatments according to the targets that's identified in their genes, or although there are a family history, we cannot find a specific variant that would, cause that specifically and show the strong familial case. So this is where our research is then also tailored a lot towards being able to use the information and confirming it before that results could be then returned to the patient. So lots of articles have been published in the process. Every time an article is published, it becomes part of the knowledge database in this pathology-supported concept. And very recently, we have also published, together with people at the National Health Laboratory Service in Bloemfontein, a very important study where a survey was also done to look at the implications for point-of-care testing. Where could this fit in to the scenario? The article has been published, so all the information is there. So our biggest challenge is really, to integrate the tumor and the germline testing results. It is very complex at times, and this is why we have developed adaptable reporting systems, so that as the knowledge grows, we could incorporate that in a patient. The doctors that is involved in the project, they can then also help us to decide whether the information that we feel as scientists maybe could add value, whether that would be valuable for their patients or maybe just cause anxiety that we might not foresee as scientists. So this development of a personalized medicine approach, where we use an integrated service and research approach. This whole process has also been described just earlier this month. A framework for the implementation of genomic medicine for public health in Africa has been published. I contributed to the Section 8, where the genetic counseling combined with point-of-care testing has been positioned as a novel approach and a new way for us to address many concerns on how do we take this forward. The informed consent for the integration of research and service delivery. Separate articles, one in Nature Genetics with many other co-workers has been published on that. The pathology-supported genetic testing approach, we're implementing it in South Africa as a case study. It is not something that is full out, applied all over, but we start somewhere to then in the end to recommend us how do we move from research to translation. On the right-hand side, you will see the process. It's a new project that has just been launched with the Medical Research Council, and eventually we would like to be able to share patient data so that this knowledge that we built can be useful to everybody. And that would obviously involve a lot of ethics approval, maybe case by case. We will have to work out the process, but also with the whole genome sequencing now available in our country and for the rest of Africa. Many of these variants of uncertain clinical significance are uncovered, and therefore we also started to build an in vitro functional study pipeline. So in conclusion, I can just say that this multi-assay pharmacogenomics platform proved particularly useful when different genetic tests are requested for the same patient over time. Some of these tests are ready for market and they are used, and that makes the system sustainable. and then other parts are then moved over to the research on the same platform. So we're focusing on diagnosis of an inherited cancer with a point of care. Should we move forward, or do we have an answer? Gene-targeted therapies, because even the most common and the most known higher-risk genes, BRCA1 and 2, they're not only involved in familial risk, they are also targets for treatment, the PARP inhibitors, where the gene is targeted, and also for prognostic value. There's all kinds of information that a single gene can give you across different domains and different timelines in a patient's life. And with this platform, it is not always easy. It takes a lot of work and attention, but you have these scientists that have a certain expertise, and they would really make that part of their research and look after what their whole life's work could bring to the party. So the affordable point of care solutions provide access to genomic medicine. This is how I see it. I don't see it as a limited test. I see it as a gateway into this whole process. One of the tests that has been validated is a drug-targeted therapy for warfarin. And I just mentioned this because we have now at least 2 patients where warfarin is interacting with their medical drugs. And then this is very important that we then look at that aspects also in the genes, if we do whole genome sequencing in the same patients, to try and minimize those risks. And then the adaptive reports is really advancing current methodologies. And we also developed a framework, and this whole process was in a way replicated in Kenya. So a framework was published in this article in Frontiers in Genetics, where we know that if we want to return research results, we first need to review the informed consent form. What did that patient actually approve? Then very carefully consider what information do we have in our database that we would be able to interpret the genetic findings that we have. Is it clinically relevant or not? Is it appropriate to return those results? And then challenges that we could not foresee at the time when we developed the consent form, at the time when the patients maybe signed that. Are there information that we could use in the report to ask patients or doctors whether they would want to know more information that we may have already in the research database? So it is really a process where lots of benefit across different disciplines. So I would just want to acknowledge all the funders. It's the Technology Innovation Agency, the Newton Fund, Innovate UK, South African Medical Research Council, CANSA, our Cancer Association in South Africa, and the Laboratory of Government Chemists in the UK. Without their technology, with the point of care, this would not have been possible. They transferred the technologies to South Africa. We can manufacture those kits now locally. It can be rolled out to rural clinics, to mobile clinics. We are looking at those possibilities. So we're really inviting people to say, is there maybe a test that you could place in a specific algorithm for your patient that could make a difference? And maybe our choices should really be reflecting our hopes and not our fears, in the words of the great man, Nelson Mandela. Thank you.